Next, I would like to invite Purva Kulkarni to present on variation in iPSC lines.
Good afternoon, everyone. My name is Purva Kulkarni. I come from the Radboud University Medical Center. And today I will be talking about iPSCs and their variations in different iPSC lines that has been reported. So we all know that iPSCs are powerful in vitro models that can help us in understanding disease mechanisms, especially in the context of patients' own genetic makeup. However, past studies have reported a lot of variance arising due to non-genetic factors, and it still remains unexplained. So together with our team, we tried to see if we can characterize this variation in different iPSC lines. And this work has been performed as a part of the PhD project of Kasper de Visser, who has performed the data analysis and implemented the whole data analysis workflow. So the multi-omics data analysis workflow, something to stress about here. So what were our research goals? Our research goals were to determine the sources of variability in iPSC lines using a number of omics layers, both looking at them independently, but also looking at the multi-omics data in an integrative manner. And the second was, once we identify this variability, can we try to still remove this or correct this variability and find disease-related markers in the individual patient groups that we used. So for this study, as you will see in this figure, it is divided in 3 stages, the experimental design, wherein we had 3 rare genetic neurodevelopmental disorders that we used, namely the N-acetyl-glucosamine, so the NANS CDG, which is a congenital disorder of glycosylation. Then we had DM1, short for myotonic dystrophy, and we also had CHD2. For these patients, iPSC lines were available, and neurons were generated using these lines. And multiple omics layers were measured using these iPSC-derived neurons. Once that was done, we decided to perform multi-omics analysis to identify the key sources of variation. And the third goal was to correct for this variation and find differential omics markers. So before we dive into the multi-omics part, it's important to understand the study design. We first started with, as I mentioned, 3 disease groups and the control lines. Each disease group had 3 patients. And for some of the patients, we had multiple iPSC lines. So we had quite some hierarchy in our data that I will show in a bit. Oh, no. Yeah, so it's also important to note here, where's the marker? Yeah. That we also had 2 isogenic lines, which were, one was a control line wherein a mutant, the CHD2 mutant, was introduced, and the second line was the DM1 patient 1 line, wherein the repeat expansion was removed to make it a control line. And also, these were all derived from fibroblasts, except for CHD2, which was derived from PBMCs. And the level of hierarchy that I was talking about can be seen here. So for some patients, we had multiple iPSC lines, which we call clones here. For example, for DM1 patient 2, we had clone 1, 2, and 3. And then these were differentiated in vitro for 7 days. These were differentiated at 3 independent time points again, which we call as biological replicates. And then for some omics layers, mainly mass spectrometry-based proteomics and metabolomics, we had each sample was measured twice, so that means we had technical replicates for these layers. So this is how the whole study design looks like. And in total, we had 44 samples and multiple omics layers, mainly whole exome sequencing, transcriptomics, methylation, targeted and untargeted lipidomics, untargeted metabolomics, which was measured at 2 different centers, one in our Radboud University Medical Center and other at the Leiden University Medical Center, and the proteomics. So as I had mentioned, the data analysis workflow was implemented, and the data was analyzed by COSPER here. As we know, as we add more and more layers, there is a huge risk of sample mislabeling and mismatch. So we used, we did a quality check for that, and we used the CNV West data to perform a concordance analysis just to check for gender integrity, sample mismatches, and once that was done, once that was corrected, we performed multi-omics data analysis. But the CNV West data was not included any further in the data integration. It was just for quality check and to correct any mismatches. Once that was corrected, we performed data pre-processing, which included multiple steps to handle the missing values, log transformation, scaling, and also quality control per omics layer. Then, since our aim was to determine the sources of variability, multiple different approaches were used, which resulted in multiple different plots. And in the view of time, I will only present a few of the major of key findings from this study. So, coming on with pairwise sample correlations, why we performed this? Because our study design was very much hierarchical in nature, so we wanted to assess the variation between the samples at the different levels that we had measured it on. So here you see 3 correlation plots. The first plot in mainly showing the red dots. On the x-axis, you will see the NANS, the metabolite levels of the NANS samples. On the y-axis, you see the DM1 sample metabolite levels. We wanted to see how the samples correlate from the different disease groups. And of course, as expected, the correlation was lower. Then we wanted to see the samples from the same disease group. So patient 1 from DM1 matched against patient 2. So this is metabolomics data that we are looking at. And the correlation was higher. Then we looked at the next level of hierarchy, wherein we wanted to see the samples from the same cell line. So biological replicate 1 of DM1 patient 1 against replicate 2 of DM1 patient 1. And again, the correlation was higher. So we were able to, in this box plot, you will see this nice hierarchy of samples, wherein the purple plot here represents the technical variables which correlated the highest. So we can safely say that the metabolite levels, also this was observed in the proteomics levels, that the technical replicates correlated the highest, and the weakest correlation was seen amongst the samples coming from different disease groups. Since we also had multiple clones from the same disease group, so multiple iPSC lines from the same patient, we also wanted to see how do the clones from a single patient perform or correlate. And what was surprising was that the correlations between the different clones from the same patient were as low as the correlations between the patients within the same disease group. We wanted to dive into this, like what could be causing this. So we started looking at the neuronal differentiation markers. So here you will see on the box plot the expression levels of MAP2, which is an early neuronal differentiation marker. On the x-axis, you will see the different iPSC lines. On the y-axis, you see the log-scaled intensity. While all other lines perform well, there was this one line, which is DM1 patient 2 clone 2, which had a different expression level as compared to the others. So on looking at the complete picture, we found that this, this particular iPSC line, DM1 patient 2 clone 2, is consistently showing lower expression for the mature markers, which are shown in green, and in the early differentiation markers shown in gray, whereas it has a higher expression level for the pluripotency markers, shown in pink here. Whereas this is not the case for many other lines that are measured as a part of the study. So this already was capturing quite some differentiation that we could see in the different omics layers. So we can say that the neuronal marker expression was not consistent among the clones within the same patient. Next, what was the main purpose here is to integrate the data and see if we can capture more variation or can we derive new findings from this. So we used the MOFA package. I think many people already presented their results where MOFA was used, so I won't dive into the details of what MOFA is. So here you will see this MOFA factors plot, wherein on the x-axis you will see all the different multi-omics layers. On the y-axis you see 6 MOFA factors that capture the variation arising from the different layers. And here you will see that the first MOFA factor captures variation or that has contributions from the lipidomics, targeted data, untargeted lipidomics data, untargeted metabolomics data, as well as a decent contribution from the proteomics data as well. Factor 2 has major contributions from the transcriptomics data, proteomics data, and also targeted lipidomics and methylation data. The rest layers, the rest number of factors, so factor 3 until 6 have major contributions from the methylation data. So we wanted to see what this means. So we started digging into factor 1, where most of the contributions were coming from the mass spectrometry-based lipidomics, metabolomics, and proteomics. So as you can see here, this is a MOFA feature weights plot, wherein you will see that some of the top scoring metabolites was doxycycline, glutamine, kinetin 9 riboside. These metabolites, this was untargeted data, these metabolites were putatively annotated based on the human metabolome database in our in-house panel. And mainly what we see here is doxycycline and glutamine. These are components of the nutrient medium. So we infer that these could be, so there could be variation in how these metabolites are transferred, metabolized by the cells. Next, kinetin 9 riboside and isopentyl adenosine. These are also crucial metabolites that are part of the cell growth pathways. Then we started also looking into the proteomics data, wherein you will see that a lot of transporter proteins are the ones that are highly enriched, or transporter proteins have top weights, as well as ribosomal proteins and also an enzyme, which is ASNS here. So SLC1A5, so we tried to determine the correlation with glutamine, and here we see a negative correlation. So we infer that these proteins have a high, are enriched mainly because they are trying to compensate for the low glutamine levels present in the cells. Also, we found this enzyme ASNS, which basically is an enzyme that is responsible for the conversion of aspartic acid and glutamine to aspartate and glutamate in the cell. So all of this, as well as these ribosomal proteins, so all of this altogether infers towards mitochondrial function as well as transport. Also looking at the lipidomics data, we saw a huge representation of multiple phospholipid classes, which are, of course, involved in mitochondrial function and cell growth. So we interpret that MOFA factor 1 reflects the differences in the expression of the transporter, ribosomal proteins, and the metabolic enzymes. Coming on to MOFA factor 2, it had higher contributions from the transcriptomics data, methylation data, and proteomics data. We found that, which was quite interesting, that a lot of the iPSC differentiation markers, so the early and the mature markers, had strong negative contribution here in the MOFA factor 2. So this is the transcriptomics data. And the same here in the proteomics data, wherein we found that after performing gene set enrichment analysis, we found that a lot of neuronal development-related pathways were being enriched here. So we inferred that MOFA factor 2 captures variation in the neuronal differentiation stage. Taking all of this information, we then moved forward to see and address the second research goal, which was to try and find the disease-related biomarkers. So for that, we used the nested linear mixed models. These models were useful and also considered the hierarchical nature of our data, so mainly the clones, biological replicates, technical replicates, and so on. So here you see this plot. I'll take you through it. On the y-axis, you will see the percentage of significantly associated omics features per omics layer here. On the x-axis, you will see all the different contrasts, so all diseases versus controls, DM1 patient 2 and 3 versus controls, DM1 versus controls. Also, DM1 was, so this contrast was separate, so we did DM1 patient 1. Separately also because DM1 patient 1 had a milder phenotype as compared to patient 2 and 3. And the rest other contrasts. So here you can see these yellow boxes. They represent the percentage of significant association amongst the omics profiles, and the numbers within these yellow boxes mainly are the absolute values of the features per omics layer. As you can see, proteomics has a higher contribution for the different contrast groups that we have seen here, and we were also able to get back the known disease biomarkers. For example, for NANS CDG, we were able to get back this MANAC6 phosphate, which is one of the known biomarkers of this disorder. Yes, and after knowing that the variability captured by MOFA 1 and MOFA 2, we wanted to correct for that variability. And for that, we regressed out these MOFA factor scores from the linear, regressed out these MOFA factor 1 and 2 scores, and then we performed linear mixed models again on our data, the corrected data. So this was able to provide us a better differentiation of some known markers, as you can see here, for, say, the MCM4, say, the L-aspartic acid. It was better after correction as compared to before correction. This also reinstantiated our trust in the workflow that we had implemented. So identifying sources of variation, and then correcting for them. So, what were our key findings? We found that the major contributors of variability were the glutamine transport, metabolism, and neuronal differentiation status, which was other than the existing genetic factors that were different among the iPSC lines. The second was that the clonal differences were visible in all of the omics layers. I haven't shown you the methylation results here in view of time, but we found based on our analysis, that this clonal differences could partly be attributed to the stochastic differences in the DNA methylation patterns, which are established during reprogramming of the iPSC lines. And third, so these sources of variability can be corrected by regression of the MOFA factors. What can we conclude from this? We can conclude that we were able to determine the sources of variability present in iPSC lines, at least some sources of variability. Multi-omics played a huge role in this. So possibly just with single omics layers, this would not have been possible. And third, we were also able to recover the known disease markers. As the next step, we still want to analyze the features that still remain unannotated but were significant, and we want to see what they mean in terms of the disease groups that we analyzed. With this, I would like to acknowledge a lot of people from within the Radboud University Medical Center, also the Chromodomain-helicase for making this study possible, and also all the centers where we performed our measurements. And thanks to you for your attention today, and I look forward for your questions.